This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 112, recorded on September 24th, 2015. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Hi, how you doing?
I'm well. How are you?
Good, good.
We're in fall here, but you're probably still the same weather, right?
Of course.
Did you get any rain lately?
We had a little rain, and there was a threat of some serious rain, but nothing happened.
We don't have much water here either. It's pretty dry. It's been pretty dry since August. But it's beautiful. Beautiful day today. Also joining us today from Ann Arbor, Michigan, Michele Swanson.
Hello.
How are you doing, Michele?
I'm doing fine, as you know.
Michelle's on her iPhone today because her equipment's not working. So if she sounds like she's in a tunnel, that's why. But I'm sure it'll still sound great. All the wisdom will come through.
I'll try.
Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Back from San Diego.
Back from San Diego, where I got to see our fellow podcaster, Dr. Schaechter. We had a nice dinner with he and his wife and
Right.
and Ray and Chris and the whole gang.
Nice. Is it the first time you saw Elio?
And in a while, and probably since the
It's been a
GM.
while.
Since the GM.
Today is September 24th, and I have to say, let me take a minute to just indulge here. Two things happened on this day. First, 21 years ago, our older son was born
Hey,
today, so that's cool.
Happy birthday.
Yeah, his Aiden's birthday, 21. He's really happy because now he can legally drink. And the other thing that happened 8 years ago today was the first episode of TWiV.
no kidding.
Wow.
8 years.
I sat down with Dixon Despommier and myself, and we recorded an episode, and it was all about West Nile virus, I remember. And that's the beginning. Yep. So thanks, everybody, for listening.
And now 1 million downloads later.
Well, TWiV has got 3.5 million. TWiM has got over 1 million too. Yeah, we're around 4 or 5 million total. So people are listening, which is great, and we appreciate it.
Mazel tov.
Mazel tov. I want to tell everyone about a couple of ASM programs for undergraduates. So if you're at an undergrad institution, you should tell them about this program. There are two of them. One's called the ASM Undergraduate Research Capstone Program. You basically will present your research at ASM Microbe. You can apply to this, and you'll get travel support. You'll participate in the Capstone Institute before you present your work. You'll present your work at ASM Microbe, and you'll get recognition in ASM publications. And then there's a second one called ASM Undergraduate Research Fellowship Program. You're going to do research at your home institution with an ASM member, and then present that work to at ASM Microbe as well. You get a stipend, travel support. You'll participate in the Capstone Institute. You'll present your research, and again, you'll get recognized in ASM publications. So these are two different programs. You can find more by emailing fellowships@asmusa.org. Two cool programs brought to my attention by Michele.
And I'd like to say that I had the great honor of interviewing a number of postdocs at the recent 55th ICAC joint meeting with the International Society for Chemotherapy. And it was really exciting to talk with these young people about the research that they have done. And we featured a number of their papers or posters at ASM Live. And we had some really cool ones from some of the presenters. From Maggie Minot and her colleague talked about antibiotic stewardship and looking at the cost-effectiveness alternatives to treating staphylococcal bloodstream infections. And we had Nishal Govinji-Bhatt talk about a contact lens case that actually reported when it contained microbes that could damage your eye. It had a color indicator in it. So she literally took it from the bench all the way to a prototype. And
Cool. That's neat.
And it's just really neat to see our future is well preserved with these young folks continuing to do research.
Yep. All right, we have two things for you today. The first is going to be a Talmudic question, and we'll send that over to Elio to tell us all about that.
Well, first I have to explain what we mean in our blog by Talmudic questions. It's a loose use of the term derived from historical, a somewhat distorted historical perception on my part. Anyhow, a Talmudic question in our terms is one where the question is more interesting than the answer. The question is, just asking the question it's supposed to make you think. That's the idea. But still, answers do come in. And this one, let me read what the question was. We said, bacterial cells are smaller than eukaryotic cells, with some exceptions. Viruses are smaller than bacteria, with some exceptions. These are just two examples of why definitions in biology, unlike in physics and chemistry, most often need to be qualified. What does this tell us about biology? That's the question. So what do you guys think? What does it tell us about biology that everything seems to have an exception?
Well,
go ahead, Michael.
I was going to say one of the answers to your question by Marco Martin is, I think, really telling. Marco Martin said that a professor of physics in college teaching him used this maxim: often constants aren't and variables don't. And teaching medical students and dental students like I do, they really just want to know the facts, and they don't appreciate that biology is this continuum that is under constant change, and that's why these exceptions can result.
Because it's changing, is that why? Because
I think it's because we're seeing evolution in real time. It's not taking place on geological scales. It's taking place in a reality that we can perceive because we're able to appreciate a phenotypic difference. I mean, the whole issue of some viruses being bigger than bacteria, and similarly, some bacteria being smaller than viruses.
I think that's a good point, Elio, because I don't think it's because things are changing. I think it's because at any given time, we don't know everything.
That's right. I think
that's true.
So, if you want to make a generalization, for example, microbiology is the study of things that you need a microscope to see. In the beginning, that may have been true, but then we discover big things that are, you can see with the naked eye. That's because in the beginning, you don't find everything. So as you find and look, you find more and more things, and there's always exceptions, and you have to revise. I mean, these kinds of statements are useful for learning and teaching, but I think they all change in the end with every field, right?
Mm-hmm.
Another one that came to my mind, Elio, was, one of the definitions of viruses used to be goes through a 0.2 micron filter.
Right. Filterable viruses, they used to be called.
Now, of course, about half the viruses would not get through that filter.
Right.
So you have to revise it. So I think that's it. Things don't change as much as what we learn is changing.
I got a variation of this question from one of our second-year med students. I gave the lecture on sterilization, and one of the practice questions asked, the best method for sterilization of serum-containing solutions for IV fluids is. The answer was a 0.2 micron filter. I noticed that many viruses are smaller than 0.2 microns.
Yep. They won't sterilize it if you consider viruses not to be sterile, that's for sure.
And so I wrote back to him that he was suffering from knowing too much.
This is also why taxonomy is difficult, right?
Mm-hmm.
You make these trees and taxonomies, and then you have to change it, and then you don't know where something new that you discover fits, and you have to go back and revise it.
let me tell you that a few of the comments that we got were by people who point out that the assumption that things are so invariant in chemistry, in physics, just ain't so. Here's what somebody said. Johann Lacerna says, stars are brighter and bigger and hotter than planets, with some exception. So, there you have it. And several people comment on that in fact, things like dark matter, Big Bang, and so forth, and for that matter, the quantum theory is something that I wouldn't say it's exceptionalist, but it's certainly not something that can be defined very readily. So, just not only is biology that way, full of exceptions, but you can make a case that chemistry and physics have plenty of exceptions as well. So that, still, I think that, like somebody called Ben Murray wrote, biology is in many respects a historical science, at least when evolution is involved. The contingent details of what actually happened matter often a lot. I think you could make similar statements about other sciences with substantial historical components, such as geology or human history itself.
But Elio, in some cases, things are constant, like the number of molecules in a mole, right? That doesn't change.
Yeah, but is that
a real thing?
No. Is that a biological thing?
No, it's not biology. I mean, in other fields, like I think biology may be one of the loosest fields.
Well, I think, how's this? I think there are some generalizations that hold. Can you conceive of a cell, a real cell, not having ribosomes? Or not having nucleic acids, for that matter? It's not conceivable,
How did
is
we
it?
get from size to no ribosomes?
Well, I'm just trying to look for
right.
I'm trying to look for laws that hold, for things that don't have known exceptions.
Well,
I mean, on Mars or on some planet, it may be that life has evolved differently, but as we know it, there are some things which have no sensible exception.
Well, would you have thought that you could have an infectious agent with no nucleic acid?
Well, yeah, no, I wouldn't have thought of it, but I wasn't totally bowled over when I found out about prions. I thought they made sense.
You're with Prishner
the fact that I didn't think of him doesn't make him that exceptional. But it's true. I mean, certainly the reverse transcriptase, for that matter, was a total surprise. I mean, surprises happen, and that's not the same as saying there are exceptions to everything.
I think the ribosome question, the nucleic acid question, that's invariant because we probably all arose from a cellular ancestor that had these things, and then everything derives from it. So maybe somewhere else, though, sure, on Mars, or maybe in another universe or another solar system, there could be very different life forms. That's fine.
Yeah, well, that's a good point. I think if we assume that life originated the way it is, perhaps not a single time, but it's a relatively small number of times, then it follows some rules. And if those rules are probably close to being invariant, because we don't know an exception. You have to go extraterrestrially to find them.
So you would say everything on Earth that we find is not surprising, or is surprising but not stunning to you in terms of biology?
I would say that. Yeah. I would say I'm ready for further surprises, but I mean, giant viruses are a surprise in recent years, certainly. But it's in the nature of things. It's not surprising, but not stunning.
Yeah. So what would stun you? Let's put it that way. Something without ribosomes, maybe?
Yeah, a cell without ribosomes would stun me. I would say that one really gets me.
Yeah.
So if somebody can dream up, how about this, listeners? Dream one up so I can feel stunned.
Something that would stun us.
Where do you put CRISPRs and CRISPR sequences in reprogramming nucleic acid?
That's just one way of using nucleic acids. I mean, that's not
to rearrange the deck chairs, as it were.
Certainly surprising. Who would have thought that bacteria have an immune system?
Yeah,
that's what it amounts to.
I'd say surprising, but not stunning, right?
Right. So it tells you that biology has a lot of surprises. That's probably the take-home lesson. Yeah. And that the surprises are still within the realm of the conceivable.
Yeah, I agree. I think it's all a continuum. You learn things and the field changes, and that's just the way the investigation is, right? There's no different. I guess all fields are like that to a certain extent, where you have an investigative process. You start out not knowing a lot, and then you learn and you change all the... we like to have guidelines to keep our thinking straight. We like to have taxonomy. We like to have constants and rules and, all this stuff. This is bigger than that. But we just have to be flexible, that's all. But I know a lot of people who are not flexible. And I don't think that's a good way to be in science, right?
Well, the people, when the Watson and Crick model was announced, there was a very notable biochemist who thought this was nonsense.
Of course, yeah. No, at every stage, there's always naysayers. The original showing that DNA can transform cells, right? People said this is not right. It can't be DNA. It's too simple. It has to be protein.
And in fact, one of our listeners, Dr. Klugman, brought that up in his ICAC lecture. He was lecturing on pneumococcus, and he paid tribute to Oswald Avery, who actually predicted that DNA was the molecule of life back in the early part of the 20th century, some almost 50 years before Watson and Crick forecasted it, because of his perception of what was going on with the pneumococcus. Dr. Klugman gave a fascinating ICAC lecture on the pneumococcus and how we've coexisted with it.
I bet. So we are left with the idea that surprises matter, but they do not sink the ship.
Right. We have to go somewhere else to do that. I don't even know if our solar system would be enough. You'd probably have to go outside of it.
Surely. No. You have to go outside, and then you got a problem of transportation.
Yeah, we can't do it.
Of information.
It takes too long. It would never happen. We're isolated here forever, probably. It's kind of too bad, but that's the way it is.
Unless we experience a Hitchhiker's Guide to the Galaxy, where they put an interstate through our solar system.
Yep. Okay, thank you, Elio.
My pleasure.
I would like to present a paper for everyone's enjoyment. This is a paper published in eLife. And lately I've been reading about fungi. So the title of this one...
Tell us why.
I knew you would ask me that.
You have red mold growing in your 21-year-old son's shower?
No, not at all. In a couple of weeks, I'm going to Germany to record some lectures for a company, and not just viruses, but bacteria, fungi, parasites, a lot of things that I don't work with all the time. So I've been reading a lot, and I read a lot about fungi. And it's just so fascinating to learn things that you don't deal with in your everyday life. So when this paper came up, I saw the title and I looked at it and it looked good. So that's why I've been reading about it.
Well, it speaks volumes about your flexibility and an elastic mind.
I don't know. Some people would say that I don't know what I'm talking about, but thank you for putting it that way.
No, you have to continue to read, and reading outside of your area, I think, is very important for helping you think about your own subject matter a little bit differently.
Well, doing TWiM, I read outside of my virus, right?
Yeah,
and that really broadened my perspective of virology. I think it made me a better teacher. But then TWiM and TWiP, I learn about bacteria and parasites. I love it. I think it's fabulous. There's a lot of threads that connect them all, obviously, and there are a lot of differences. But, a microbiologist by training should be able to understand all of this. So I don't have a problem with it at all. I don't look at it as a chore to read other things. This paper is called Phytotoxin Production in Aspergillus terreus is regulated by independent environmental signals. Is it terreus or terreus?
I like terreus, but I think most people in this country would say terreus.
Terreus. And it's published in
And there's a
eLife.
rule, by the way, there is a rule about pronunciation.
Yeah.
I looked it up once. What's the proper pronunciation of Latin? Well, the proper pronunciation of Latin turns out to be what most people in a given place are comfortable with. So terreus is okay, terreus is okay.
Okay. Anyway, this is by Gressler, Meyer, Heine, Horchansky, Hertweck, and Brock. They're from Jena, Germany, and Nottingham, United Kingdom. And this is all about Aspergillus, which is one of those fungi that grows in the soil everywhere, and it makes hyphae and mycelia. And it's quite ubiquitous, and it does very cool things. So this paper is all about what are called secondary metabolites. So organisms make all sorts of metabolites, small molecules that they either need for their growth, those are primary metabolites, or small molecules that they're not essential, but they make their life easier. They help them survive, they help them to compete, and they help them reproduce. So this fungus called Aspergillus terrein.
Before
Yes.
you go into it, can I bring up a point which I think very, very interesting, namely that by using techniques of mass spectrometry, people like Peter Dorenstein have been able to show that bacterial colonies emit extracellularly hundreds of compounds.
Yeah.
So this is given a new dimension
Sure.
to this idea of what is it that bacteria and fungi make. It's much greater a repertory than people had thought, and it's going to alter our view of a lot of things.
And it brings up another Talmudic question. What benefit do these secondary metabolites offer the microbe?
Right. And that's part of what this paper is all
That's
about,
the question,
of course.
this paper, right.
Now, this Aspergillus, again, it's a soil fungus, is known to make secondary metabolites, and some of them we actually use. For example, one of them is called lovastatin. It's marketed by Merck as a drug called Mevacor. And you should know that this was a blockbuster drug for Merck. It lowers your cholesterol.
And I'm on it.
You're on it? This is a product of Aspergillus terreus. All right, isn't that amazing? So they discovered it, natural product, and they actually produce it in fermentation, and they sell it as a drug. And I have to say, I benefited from it for years. My wife worked at Merck for 27 years, so some of that Mevacor money certainly went my way. Another secondary metabolite that this fungus makes is itaconic acid. And this is, again, produced commercially by fermentation. I don't actually know if Mevacor is produced by fermentation or not. I shouldn't have said that, but it is.
Probably.
I think so. Probably not by chemical synthesis. Yeah. It's easy to do it. Anyway, itaconic acid it's used to make styrene and acrylic latex paints.
Oh, we need acrylic latex paints.
Yeah, everybody knows that, it's really easy to clean up a brush that you just used to paint the wall with acrylic paints. It's water soluble. So this is a fungal secondary metabolite that makes our life easier. I mean, this is just one of thousands of examples of how the microbes help us. It's just amazing. Anyway, another interesting side point here, we all inhale spores. If you live near dirt, you're probably always inhaling spores of Aspergillus of all sorts, and most of us have no problem dealing with them. But of course, if you're immunocompromised, if you have AIDS or you're immunosuppressed by virtue of a genetic defect, or you have an organ transplant, you can get very sick from inhaling these. They grow inside you. Now, terrein, a terrein produces another secondary metabolite, which is a subject of this paper, and it's called Terrein, Terrein. And
comes from terra. Terra is earth.
Terra is earth, right? It's an earth fungus. Terrein is produced by this fungus that lives in the earth. This secondary metabolite, this terrein, is a pathogen of potatoes. It makes lesions on potato leaves, so we call it a phytotoxin. And it also has antifungal properties. And
not against itself.
Not against itself, but against other fungi. It makes sense, right, in the earth. If you want to get rid of your competitors, you make an antifungal, and you say, leave me alone in my little 1 foot circumference or whatever, and get rid of competitors.
And that's the other thing you may wish to mention is that in addition to making terrein, it probably makes the antidote to terrein as well for itself.
Might, yeah.
Good point.
Good point. So very recently, the genome of this fungus was sequenced, and the authors said they found the gene cluster that's responsible for terrein synthesis. They found a whole bunch of biosynthetic genes. They found the promoter. And so they said, okay, now that we know this is here, we'd like to know what this molecule does. So I'm going to tell you about some of the experiments they did. The molecule, by the way, is a small organic molecule. There's a 5-carbon ring. There's an oxygen off of it, a couple of hydroxyls, and then a chain of 1, 2, 3 carbons. And that's it. It's a pretty simple molecule.
Yeah.
It has lots of properties. So they have the gene cluster. So that means more than one gene is involved in the biosynthesis. And there's one gene which encodes a protein called terrein synthase, or TerA, and it's got a promoter. So they take the promoter, they hook it up to the beta-galactosidase gene. That way they can measure the production or the expression of the TerA gene by looking for blue color, because beta-galactosidase will convert a precursor to a blue color. It's a lot easier than making antibodies to the TerA and then doing Western blots or whatever. You just measure blue color. So a lot of the experiments are doing that. So they make a strain where they've inserted beta-gal into the TerA, behind the TerA promoter. So what they find in a number of very nice culturing and biochemical experiments, they find that if they grow the bacteria on glucose, it induces this promoter.
Fungi.
Sorry, did I say bacteria?
You did.
I'm so used to talking about the fungus.
What are you, antifungal?
No, not at all. I love them. I think they're great. They're so cool. They're beautiful too. And as you know, Elio, they can be miles in size, right?
Of course.
So glucose induces this promoter. Nitrogen induces it even more. And they know that this terrein causes lesions on fruits, as I told you. So they try cultivating the strain with the beta-gal in it on apple, peach, banana, and carrot juice, making me hungry. And this induces the promoter even more. So next, they infect fruits. They infect bananas, apples, nectarines, and they use the wild-type fungus or one where they've deleted the terrein synthase gene.
By the way, it's really neat. The picture of the banana being infected at different dilutions looks just like a petri dish.
It's great. It really is
amazing.
Great. This is open access. You can all take a look at this. So if you delete the terrein synthase gene, there are no lesions, of course, and there's no terrein made. But if the synthase gene is present, you get lesions on the banana, and they can detect terrein production. So terrein production is associated with lesion formation. Now, in other experiments they did, they found that when they added casamino acids to these cultures, it stimulates this TerA gene over glucose alone.
By the way, that's like a mesh of amino acids that comes about if you take casein.
Right.
From milk, which is a cheap source of proteins, and hydrolyze it, you get just a mixture of peptides and amino acids. And they call that's a commercial term, case amino acids, which has been around in microbiology forever.
Right.
If those of you who are listening in out there, you can think of it as Vegemite for bacteria.
Right.
So that said to them, well, maybe an amino acid is important. So they do a number of experiments. I don't have to go through them with you, but it turns out there's one amino acid, it's methionine, that induces the synthesis of TerA. And the methionine, of course, is a sulfur-containing amino acids, and when you start a protein, you often start it with methionine. Even if you take...
Methionine, by the way, is really an oddball amino acid, isn't it? It really plays a lot of special roles, not just being part of being protein. So it's really interesting. I'm not surprised the methionine popped up here.
Now, if you add glucose plus methionine to cultures of the fungus, it still induces the terrein gene less than banana juice. So what is in banana juice that makes it such a good inducer? So what they say is fruit juices in general have a high carbon to nitrogen ratio. I didn't know this. Okay. So a lot of carbon to not much nitrogen in fruit juices. So they're saying maybe nitrogen is limiting. So they do it.
You sort of knew that just by tasting fruit juice.
I don't know what nitrogen tastes like. Do you?
Amino acids.
It's not sweet.
Fruit juice is not sweet?
No, nitrogen's not sweet.
Amino acids are not sweet.
Yeah. If you had a lot of amino acids in fruit juice, it would be less sweet, you're saying?
Yeah, it would have a bitter type taste. Think of aspartame when it goes bad, the artificial sweetener. If you've ever had your Diet Coke go stale, you're effectively eating the breakdown of the two amino acids, the phenyl. Yeah, when they skunk.
Interesting. Well, people in my family, they buy these huge containers of protein and they make drinks out of it.
Oh, the whey?
I don't know what it is, but it's protein supplement, basically. My son, he's a bodybuilder, and he drinks tons of this stuff to build muscles, I guess. So I guess
whey.
He would know.
It's probably whey protein.
Anyway, so they do a bunch of experiments which show that, in fact, under low nitrogen levels, limiting nitrogen, you get an induction of terrein, even though it's delayed. You don't get it initially. So basically, when you grow on a source, a plant juice, initially there's no induction, but then as growth continues, the nitrogen becomes limiting, and then you induce this thing. So starvation of nitrogen seems to induce the terrein gene. So far we have glucose, methionine, and starvation of nitrogen are all inducing this terrein gene. And remember, we want to find out what's making terrein, what conditions lead to it, and that can help us figure out what it's doing. Now, so in fruit juice, nitrogen is limiting. If you add a nitrogen source to banana juice, for example, you reduce the induction of terrein, and that makes perfect sense. And the same thing when you infect bananas, you can add a nitrogen to the banana, and it reduces the induction. Okay, so sugars, methionine, limiting nitrogen. So how is nitrogen starvation and methionine detected and passed on to the promoter? So that's a key question here, right? So methionine induces it. How does it do that? So again, they go back to the genome sequence and they identify proteins that are involved in nitrogen sensing, stress response, control of amino acid synthesis, and they pick 4 or 5 of those that look interesting. They delete each one from the fungus. So you can do genetics here and delete genes specifically. And they look at the growth of the strains under different conditions. So this gets a little complicated here, but I'll try and simplify it for everybody, not just non-scientists. So there's a gene called AREA, or area. This encodes a global nitrogen regulator. When you delete this gene, the fungus doesn't grow very well. It has growth defects. It can only grow well in medium with nitrogen-rich amino acids or urea or ammonium chloride, all sources of nitrogen. Okay, so AREA is a nitrogen regulator gene. There's another gene that they got interested in is called ATFA, alcohol, tobacco, and firearms. No, sorry. ATFA.
You had to go there.
I had to do that?
You had to do that.
Yeah, ATFA. Because a transcriptional regulator, that is, it's a protein that regulates the transcription of a variety of genes. It's responsive to stress. So when cells get stressed, they induce a lot of genes to do different things to kind of alleviate the stress. And so this is a regulator that's involved in that. When they delete this gene, it prevents the synthesis of terrein. Okay? So that's kind of a nice fortuitous discovery there, because you wouldn't have predicted it, I guess. Both mutants, deletion of AREA and ATFA, they make very little terrein when you grow them in glucose with limiting nitrogen. So these are conditions where you should make a lot of terrein. Make a double mutant with both AREA and ATF knocked out, no terreins produced at all. So both genes you need to have terrein production under conditions of limiting nitrogen. Okay, next step in this story. TER is a transcription factor that activates genes within the cluster of the terrain synthesis genes. If they take the gene for TER and they overproduce it in an area ATF double mutant, you make terrain. In other words, somehow area and ATF are regulating production of this transcription factor. So there's a signaling pathway in there. We don't know what it is. They haven't sorted it out, and I assume at some point they'll do it. But there's a signaling pathway that goes from area and ATF to this transcription factor that regulates the terrain synthesis genes. Okay. So there's a methionine-dependent signaling cascade that's talking to these genes, and there's a nitrogen-dependent signaling pathway that's talking to these genes as well. All right. Then the last part of this story is iron.
Of course.
It always comes down to iron, right? We always get iron in this story. They wanted to know what trace elements might also be involved in inducing. They did a series of experiments where they add certain trace elements, and a lack of iron and only iron and no other trace element also can induce terrein. So terrein, again, is the terrain synthase gene that we've hooked up to a beta-gal, and a lack of iron turns it on. Now, previously, other people showed that terrain has antioxidative properties. So they asked, can terrain reduce ferric, which is 3 plus Fe, to ferrous, 2 plus iron? Okay. And in fact, they find that terrain can do this, but the pH optimum for the reaction is between 3 and 4. And
so they're getting a little help from the acid of the medium.
Exactly. And as they grow these cultures, that in fact it goes from a neutral pH to a pH 3 under iron limitation, and that activates the ability of terrain to change ferric to ferrous. Every time I see ferric and ferrous, I think of ferric fang. I mean,
no, he's got it funny. When you ask him, do you have a cousin called Ferris? He says, yes, but he's unstable.
Yes, I was going to mention that. It's great. His mother must have been very clever to call him Ferric, right? What a great Ferric and Ferris. He's very unstable. So basically, when terrain, I'm getting terrain and ferric confused now. When terrain is produced under low pH, it could improve iron availability because ferrous iron is more soluble and could be taken up. There's a ferrous iron transport system. So they're hypothesizing that maybe this change from ferric to ferrous is helping to make iron available. Now, as everyone who listens to TWiM should know, microbes make siderophores to bind iron and take it out of the environment and pull it into their cells. And in other fungi, it's been shown that there's a gene called HAP-X, which is a transcriptional inducer of siderophore synthesis, and it's regulated by iron levels. So in their Aspergillus, they find that HAP-X exists, and it regulates both siderophore production and terrein production. So when you starve for iron,
that's neat. That's really neat.
You get induction of the terrein operon by HAP-X in response to iron starvation. So here's what we have. We have two iron acquisition pathways. We have siderophore-based and reductive iron assimilation. If you go from ferric to ferrous, then you have a ferrous uptake mechanism, okay?
It shows you how important iron is to this microbe.
Pathways. That's right. Now, the SID-A gene in this fungus is essential for siderophore synthesis. Without iron supplementation, a mutant deleted for this gene grows poorly. But when you add terrain, even in iron-limiting conditions, it can grow. So they think that while siderophore iron acquisition is dominant, terrain can also support iron acquisition by doing this change from ferric to ferrous and allowing the ferrous iron to be taken up. So what does this all mean for the biology of the Aspergillus? Okay, terrain inhibits plant seed germination. It makes lesions on the surface of fruits, and it reduces ferric to ferrous ion. The idea is that this makes the fungus competitive in the field. All right. The production of it is regulated by three signals: methionine, nitrogen limitation, and iron starvation. So the way they put it is in nature, this fungus is growing in the soil. There are lots of nitrogen-fixing bacteria in root
Also,
nodules.
being phytotoxic, it may make vegetable matter more accessible to the fungus.
That's right. Absolutely. That's one of their hypotheses, that the phytotoxic properties help it, get nutrients from other plants by destroying them. But they say the nitrogen limitation signal may be because, the nitrogen-fixing bacteria are removing nitrogen from the soil. So terrain synthesis is therefore stimulated, and that kills the plant and releases nitrogen so the fungus can have it, right? Makes sense? Nitrogen low, turn on terrain, kill the plant, release the nitrogen. Plant roots also give off methionine, they say. I'll take their word for it. So if there's a nearby plant, they sense the methionine, terrain is made, the plant's killed, and the fungus can benefit by it. And then what about iron? Siderophores bind ferric iron, right, Michael? Not ferrous?
Yes.
Right? So when terrain is produced, it reduces ferric to ferrous ion. Maybe this prevents siderophores from other organisms from being able to bind the iron. So the fungus
it's a competition.
Yeah.
It's effectively tying up the valuable resource.
Exactly. So the terrain changes the iron to a form that can be taken up by the fungus, the ferrous form, but it cannot be bound by the siderophore. So it's a competition. This is a hypothesis, of course, and I guess they can test it at some point. So anyway, basically, they've given us a bit of insight on how terrain is induced, what conditions induce it, and basically show that it really is needed probably for the fungus to acquire nutrients that maybe its competitors would otherwise have. So I think it's a lovely paper. It's very long and lots of experiments, but lovely combination of biochemistry and microbiology and reaching these conclusions.
And it's all done with the old beta-gal assay, where they use simple colorimetric methodology, where they can now drop the beta-galactosidase gene into a fungus that normally doesn't have it in order to measure activity. And that's the beauty, is they actually can measure activity and figure out what genes are firing where when they drop this reporter cassette into these various constructs that they have. So it's a beauty of molecular biology in concert with the biochem and as well as the mycology.
So Michele, who unfortunately has dropped off, spoke with Marcus Gressler, the first author on this paper, and she has some really cool things. Marcus was born and grew up in Arnstadt, Germany. He says very early on he got in touch with microbiology since his parents liked to collect mushrooms in the Thuringian forest around his small town. How about that, Elio?
Well, it's true for so many people in Europe. If you ask most anybody who grew up in Germany, Poland, Russia, Italy, did you ever go mushroom hunting? He says, of course.
Yeah.
My grandmother would take me. She knew which ones were good to eat.
Right.
That's an almost universal answer.
But here in the U.S.,
nice, but not surprising.
Here in the U.S., not so much.
Not so much. We're mycophobes.
Mycophobes.
I like to say people in the world can be divided into mushroom pickers and mushroom kickers. And Americans tend to be mushroom kickers.
That's funny.
Unfortunately.
I have an entire front lawn full of mushrooms. It's been raining here for the last
Take a
month.
picture and send it to me.
I'll take pictures and send them to you.
Okay.
Mushroom kickers and mushroom pickers. Anyway, Marcus studied biochemistry, molecular biology at Friedrich Schiller University, which is in Jena. I've been there. And he worked on Aspergillus terreus, which we've talked a little bit about. He mentions how it led to the making of $1 billion drug, lovastatin. Got his PhD in the same group under Matthias Brock, who's the head author on this paper. And he wanted to study terrain. And he says one of the interesting things is that you can make it in grams. It's very easy to make tons of. And
the fungus makes it in grams.
Yeah, the fungus makes it in grams. And it's formed during growth on fruit juice. He says, I still remember the smiling face of our technician entering our 30-degree centigrade cultivation room and deeply inhaling the flavor of banana or peach juices. However, her smile quickly changed to a disgusted face when she looked at me grinding a decaying dark brown banana that was infected with the fungus 10 days before.
Yes.
After finishing his PhD, he started working on the biochemistry of oligosaccharides from Aspergillus fumigatus at the Institut Pasteur. And in the future, I guess that's what he's doing now. And in the future, he wants to work on the development of drugs against pathogens or cancer in a pharmaceutical company. He says, although basic research allows me to follow my own sometimes crazy ideas, I currently miss the direct link between my research and the benefit for the patient. Anyway, lovely work. Really
laudable
lovely
goals.
Lovely work. Nice job, Michele. Thank you very much for speaking with him. We have a few emails to read here. First is from Steve. Oh, this is so apropos. He sends a link to an article. Mysterious fungus killing snakes in New York and at least 8 other states. One for Elio if he's not onto it already. Amphibians, bats, and now snakes. Have the fungi just declared war or what? Steve is in England. So the fungi are killing bats, white nose syndrome. They are killing amphibians and now snakes. Do you know what's going on, Elio?
No, I don't. But it's serious. It's bad stuff.
Why all of a sudden,
It's a long name, maybe Michael remembers the name of the fungus.
It's not It's not a variant of Stachybotrys, is it?
Which one? The snake one?
No, it's not. No. It's something like it, but I've forgotten.
It's Ophidiomyces. I'm just curious why, in the last few years we've had this increase in killing of these animals by fungal diseases. Fungi like to grow at lower temperatures, right? Close to the earth and in the earth.
That's right.
And amphibians certainly fit the bill.
And they're in the soil, so.
And bats in the caves, reptiles. But why all of a sudden the increase, right? Some people think it has to do with, chemical toxins and so forth, but nobody really knows. Interesting idea.
Right.
The next one is from Petra, who writes, Dear TWiM, thank you for your excellent discussion about graduate and postdoctoral education in TWiM 108. I enjoyed the conversation about career options for people with advanced training in microbiology. I earned my PhD in microbiology and did postdoc training before taking my current position as a research scientist at 3M Company. As a scientist doing research in the industry, I don't think of my career path as alternative, although I often hear that term used to describe non-tenure track careers for scientists. It would be a step in the right direction for advisors in graduate programs to start using the term careers instead of alternative careers when talking to students about options for the future. The use of the word alternative to describe non-tenure track career options seems to be particularly pervasive in academic departments specializing in the biological sciences. As biologists, let's start a movement to catch up to our colleagues in chemistry and physics departments where moving to non-tenure track positions after graduate school is a normal and valued option. Many thanks for a great show. Listening to TWiM is a great way for me to stay current on a wide variety of topics in microbiology, which is not only fun but part of my job. I totally agree. I don't like to use alternative career.
I think she's
anymore.
right.
And you're right.
It's more pejorative. It's really pejorative.
We've already stopped doing it here at my institution. Probably about 5 years ago. It's
good for you.
Anthony sends a link to an article about how diphtheria antitoxin was delivered by dog sleds up in Alaska. This is a story I've heard before.
Yeah.
It's pretty cool. So
it's the one I tell the dental students.
Yep. And Suzanne writes, I was so confused when I first heard you talking about this. And she's referring to the last podcast I think we did where we talked about a paper where they reproduced an old English remedy. Right?
For the staphylococcal, for treating staphylococcal with copper pot and the eye of newt. And
right. It sounded so familiar. I was sure you'd mentioned it before, but it sounded like it was new to you. After a bit of thought, I wondered if I'd heard of it during a class I took on Beowulf last spring. I checked the discussion board archive, and there it was. Someone had linked to an article about it on newscientist.com. What a cool overlap between online communities. If you're still wondering, the language spoken in England back then is now either called Old English or Anglo-Saxon. Beowulf was written in that version of English, and it's nearly unintelligible today unless you spend time learning the language. But there are still familiar words here and there. Chaucer wrote in Middle English, which we English speakers can still understand somewhat without a whole lot of help. My favorite thing about Old English, really about pre-printing press English, is that there were dedicated letters for our TH sounds. One was like a cross between lowercase p and b, which looked kind of like a mouth with a tongue sticking out, which makes perfect sense. Next one is from Witama.
By the way, no, but I appreciate this connection to think that microbiology and literature and history can work together. It's just lovely sound to my ears.
It's great. Absolutely.
Well, what happened is that paper came out, if you recall, came out as an abstract at a meeting. And what we discussed on the last TWiM was actually the abstract being converted into a manuscript.
Mm-hmm.
That's right.
So what made the news was the abstract, and what we discussed was the final manuscript with all the data.
Right.
So it's fantastic.
Next one's from Wintama, who writes, Dear TWiM hosts, my name is Wintama. I am an honors student in microbiology studying in northeastern Australia, where it feels like a pleasant 18 degrees Celsius. For your information, honors is a year of postgrad research that can bridge you into a PhD without completing a master's degree. Anyway, my question is about Taq polymerase. My girlfriend Anna is currently doing some TA cloning, and I was surprised to hear that the Taq polymerase enzyme adds an A overhang to the ends of the PCR product, making it perfect for this cloning method. I was wondering what biological significance this function has. I guess the first step would be to insert a polymerase lacking this function into the bacteria and seeing what happens, but I'm a little busy with my own project on ranavirus to take it on. Thanks for the great podcasts. So, Wintama, how would you make a polymerase lacking specifically that function? I think it would be really hard because it's probably just part of the polymerization activity of the enzyme. So Taq polymerase and a lot of other bacterial DNA polymerases can add a single non-templated nucleotide to the 3' end of a blunt DNA. Function unknown as far as I know, because these polymerases are typically copying circular DNA genomes, right? So when would you have a blunt end? Maybe when there's DNA damage. Maybe it's a signal for DNA damage of some sort. But I did not find anyone who knows what the function is. Do you know, Michael Schmidt?
No. Like you, I think it's probably to signal either the rec system, principally rec A, that something's gone amiss with the replication process, and to signal recombination or at least repair.
Right. All right. Matt writes, Hi, Tweet team. Absolutely love your podcast. I'm a public health specialist coming to the hard sciences late in life and enjoying it immensely. Thank you for all you do communicating the wonder of science. That said, I think you blew it on the public good explanation in TWiM 111. The reason public goods are fundamentally different than private goods is that unlike private goods, the producer of the public good gains no advantage vis-a-vis his peers. If a Pseudomonas bacterium developed a mutation that allowed its siderophores to differentiate gallium from iron, that benefit would be diluted across the entire population of the colony. Everyone, including the mutant, would encounter the same environment filled with a majority of gallium-bound and therefore toxic siderophores. There would be no individual survival advantage to the individual that developed the mutation, thus no selection pressure. Now, if there was a mutation that allowed a cell to make use of the gallium as if it were iron in its cell processes, that would be a different story, since that would be an individual advantage and would be selected for. But given the diversity of uses of iron in various metabolic pathways, the likelihood of an individual mutating to accept gallium in all the places where iron is currently used is essentially non-existent. So the public versus private does matter. Keep up the amazing podcasts. My dog loves your show because he knows when I put on the headphones, he's going for a long walk. Yeah, we did blow it. That's a good explanation, right?
We may have partially blown it. I think he's right.
He's got a good explanation.
I think we dealt with it a little bit superficially, and I'm sorry.
Yep, I agree.
Might as well apologize in
advance.
Peter writes, Dear Twimmers, I'm Peter from Falmouth, UK, not to be confused with its no doubt sunnier namesake in Massachusetts. We're somewhere about 16 Celsius, that's about 61 in your currency, with showers, and autumn is starting to tell. I'm just writing to tell you to comment on the evolution of resistance to public goods-based antimicrobials. So again, it's another letter about public goods. I'm going to first run with the assumption that there is a mutation somewhere in gene space that allows the siderophore to exclusively pick up iron. Then the problem with public goods for the bacteria is that the individual bacterium that gets that mutation isn't necessarily the one that gets the fitness benefit. And he goes on to talk about how other bacteria could get the advantage in the population, and therefore there's no selective advantage. I suppose most of the places we're going to be medically concerned about P. aeruginosa are going to be places with a bit of spatial structure, but still the fact remains that compared to a completely privately operating mechanism, a public goods-based antimicrobial will be less prone to resistance simply due to the who gets the benefit of the mutation idea, though it seems to me that resistance would nonetheless eventually evolve given sufficient time and selective pressure. That's all, except I just want to say that I have a lot of love for TWiM and TWiV and what you guys do. Over the last 5 years of tuning in, you feel like familiar characters, the sort that I might meet regularly in the local pub and chat the night away with. And as a PhD student who never does enough reading.
Is he buying?
Yeah, sounds like it, but a PhD student may not last long. As a PhD student who never does enough reading, I cannot say enough what an amazingly useful tool you guys are providing. Multiple lifetimes worth experience poring over the latest bleeding edge research with such wonderful insight. What an inspiration. Cheers all, and here's looking forward to 112 and beyond.
Nice.
Well,
Thank you.
Wonderful insight, except when we blow it, right?
Right.
But we must not have blown it too badly because it was sufficiently provocative that we got 2 great responses that set the listening world on track.
And we have one more, so let me make that
Okay,
the last
one more.
because another one. Kevin writes, Dear Twimmers, I am writing to help clear up some confusion as to why resistance might be hard to evolve if public goods are targeted. In 111, Elio talked about gallium intoxication of siderophores, and a proposal was put forth that since the siderophore is extracellular, it would be hard to evolve resistance to gallium. Vincent said it is the atomic mimicry of gallium and iron being so similar that is important. Loss of gallium binding would almost certainly cause loss of binding to iron. Elio replied that both the extracellular nature and the mimicry are important, but why extracellular matters was perhaps not so clear. I think the distinction is not extracellular versus intracellular, but rather that the siderophore is a public good that has impacts on a population. In contrast, mutations that may produce an altered siderophore occur in individual bacteria. What this means is that mutant bacteria that produce a siderophore that distinguishes iron from gallium does not receive the full fitness benefit of that mutation. Instead, the whole population gets it. In this manner, fitness advantages from a mutant are distributed to a community instead of one bacterium. However, in my opinion, this is still not necessarily a satisfactory explanation as to why resistance would not develop. Even if the whole population receives the advantage from one individual, the mutant bacterium is among that benefited population and will receive some benefit. The counter-argument would be that by spreading the fitness advantage among a population, the fitness advantage is so diluted that there is virtually no selective advantage conferred to the mutant bacterium. Personally, I think targeting public goods such as a siderophore may slow expansion of resistant alleles in a population, but not prevent them from occurring. So that's the second one who says that.
Yeah.
It's interesting to conjecture that diffusion properties of these public goods to a large population or a relatively small population of bacteria may impact the rate at which natural selection occurs. Thanks for your great work. Look forward to the next episode. But if there's this idea that altruistic mutations can also be selected for as well, right? Mm-hmm. So I think if you had a mutation that benefited the whole population, that would eventually dominate, and maybe you could get resistance. Maybe it would be slow, as these two say, but maybe you would get it eventually anyway.
Yeah, I think they nailed it with the whole notion that it's spreading amongst the population, so the selection pressure is being diluted.
It is, but what if it helps the whole population? Wouldn't they then all have that mutation eventually in them, and that would then confer some resistance? I mean, maybe it takes longer, but
it's the issue is most of the really nasty infections are pure culture type activities rather than a mixed infection, unless you have some sort of wound. And so I think here it would actually control the pure culture type infections that are most challenging often to control, with especially in the pseudomonads and acinetobacters that are seem to be resistant to all our ethical drugs.
Right. All right. Thanks for your letters. This is TWiM
That was
112.
terrific, in fact. What a collection of letters. Wow.
Yeah, we're very lucky here at TWiM, and you can find this episode, all the others, at iTunes at microbeworld.org/TWiM, and you can use your iPhone or Android device to get the podcast as well. And if you have questions or comments, please send them to twim@twiv.tv. No comment is too big or too small. We take them all.
That's right.
If you want to learn something that you don't know about, send it to us. One of us will figure it out. If we can't, we got a lot of people listening who can. I'm sorry that Michele Swanson couldn't make it. So, Michele, get your equipment there in order for the next time, please. And Michelle's at the University of Michigan in Ann Arbor. Elio Schaechter is at Small Things Considered. Thank you, Elio.
My pleasure. Wow, this was something.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, Vincent.
And I'm Vincent Racaniello. You can find me at virology.ws. I want to thank ASM for their support of TWiM and Chris Kandayan and Ray Ortega for all their help. The music you hear on TWiM is composed and performed by Ronald Jenkins. Check out his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.